I am pleased to be hosting Dr. Maria Diehl to discuss her recent PLOS ONE Lab Protocol paper entitled Modifying the Platform-Mediated Avoidance Task, a new protocol to study active avoidance within a social context in rats, and the associated detailed protocol in protocols.io. Maria is currently an assistant professor in the Department of Psychological Sciences at Kansas State University. She studies the neural mechanisms of fear and avoidance in the context of social behavior using a wide array of modern neuroscience techniques. After discovering an inhibitory signal in the prelimbic prefrontal cortex that is essential for platform-mediated active avoidance, Maria mapped bidirectional outputs of prefrontal circuits that guide active avoidance. Currently, her lab has modified the platform avoidance task in several ways to understand how social cues affect avoidance learning and how the neural circuits shift under these conditions. Maria has received a corporate research project grant from the Cognitive and Neurobiological Approaches to Plasticity Center at K-State to study the effects of social interactions on the neural circuits of fear and avoidance. Maria, thank you so much for carving some time to discuss this paper and protocol with me.
Thank you. Thank you so much for inviting me and having this opportunity to talk about it.
So to kick off, can you briefly explain what the protocol is about, how you use it, and why it is relevant?
Yeah, so we use this protocol to train our rats in the laboratory to perform an active avoidance behavior. So the way we do that is the animals first learn that a tone will predict a foot shock, and they kind of discover as they're progressing through the task that they can avoid this foot shock by stepping onto a platform. At the same time, they have already learned that they can press a lever to get a sucrose reward. So we have these little sugar pellets that they can press this lever for. But when the tone comes on, they have to stop pressing for that sucrose reward to avoid the shock and get on the platform. So there's this conflict that arises where they have to kind of realize, oh, I can't be pressing anymore because something bad's going to happen. I need to take action. So for many years, there was this task that was developed kind of in modification of an auditory fear conditioning task. And we were really interested in how we could use this task to understand how social information might be used to understand how something dangerous in our environment can affect, how we take action and deal with that danger. So for a long time, we had a solitary version of this task where the animals were learning on their own. They're in their own operant chamber. They're pressing the lever for sucrose, and they learn this avoidance behavior. But I was really interested in how we could incorporate a social partner and see how the behaviors might change. So in this task, we're really interested in looking at freezing behaviors, which is a measure of fear, also how their pressing changes when the tone is present versus not, and also how much time they spend on the platform as a measure of this avoidance behavior. So in this protocol, we have all the information someone needs to set everything up, all the materials, all the equipment, and the software that we use to track the animal's behavior during the task. We think this is really important for trying to understand learning and memory processes that occur during active avoidance. And using a rodent model can help us, uncover the neuroscience that gets behind the learning and memory processes. We think this is really important for trying to understand what happens in different kinds of human diseases like PTSD, OCD, and anxiety, so that we can see what are the neural circuits that might go awry in these behaviors and how we could use that to develop future treatments for these kind of diseases.
And to further understand the significance of this protocol and paper, can you elaborate on what was the state of the art? weren't there any protocols to study fear in a social context before you developed the protocol and published the paper?
Well, most of the protocols that we, would find, they weren't really published in a formal way. So, you might go to a conference and talk to someone at their poster or after their talk, to get more information on how they set up the task. What did they do to overcome these different kinds of challenges? And if that person was also trying to set it up, they might have additional questions about different details. So before this protocol was published, we really hadn't had any kind of formal publication on how the platform-mediated avoidance task was set up. Just kind of reading details in other publications using those tasks. So I really wanted to have kind of a formal protocol that outlined, all the things that we use, all the different supplies we buy. We may not use specific equipment from different vendors, but we might tweak some things and, kind of make some things on our own. That kind of information is really important for other researchers if they want to replicate this kind of work.
And from what you learn in these less formal conversations that you have with colleagues who use similar approaches, can you summarize what are the unique advantages that your protocol offer in comparison to alternative approaches that other people use in the field?
So I think the main focus that I'm really interested in is that social aspect. So this protocol is really helpful in understanding, what are some things you need to consider if you're really interested in looking at social behavior or social interactions between rodents, because that can be a big challenge. There's a lot of things that happen when you introduce social factors that you can't control for. You can't control, what one animal is going to do and how the other animal responds to that animal. So we try to put some things in place to make it a little bit easy for the researcher to track the animal's behaviors in separate ways. We have 2 different cameras so we can independently track the animal's behaviors. And then, have it in a semi-realistic setting so that there's a good balance of tracking what each animal is doing versus allowing them to, freely behave as they would in a semi-natural setting.
I would like us now to go into more detail about the process of developing and standardizing the protocols. What were the highs and the lows? And specifically, is there anything that you tried when you were setting up and optimizing the protocol that didn't work and didn't make it to the final version of the protocol?
Yeah, so something that's related to this and a question I would get a lot is why is there a barrier? So if you look at the paper, there's a picture of in one of the figures where we have the 2 animals who learn the avoidance task together at the same time, but they're separated by a plexiglass perforated barrier. So initially, we didn't really want the barrier. We wanted, something as realistic as possible where you have the 2 animals, they can interact with each other, they can crawl over each other and do whatever as they would in the real world or in the wild. But realistically, trying to measure, their avoidance behaviors and other behaviors that happen in the task, it wasn't very practical. So to kind of overcome that aspect, we introduced this barrier so that they could still see their rat partner. They could even smell and get olfactory cues from their rat partner, and still have that chance to press the lever to get their sucrose reward and pursue that motivational reward, or have their own access to their platform and not have that issue of the animals possibly competing, eating each other's food, pushing each other off the platform and things like that. So I think this was a really good balance of having that ability to see what each animal is doing, but also having them able to interact in a limited way to see what those social cues they might be using to do the task.
So the plexiglass barrier separating the 2 animals is critical for practical purposes. Is there any other step or part of the setup that is critical as well and might be easier for others to make a mistake on?
Yes, so I think these are more general when you're working with rats and if you're doing a procedure that takes multiple days. So I'd say the most important thing is for researchers to make sure they're doing the same thing every day and treating all the animals in the same way. That will help prevent any, additional variability that would come into your data. So, in addition to having to train the animals to learn how to lever press to get the sucrose, that takes a few days. Then we do the PMA training, which takes 10 days. So it's really important that every single day, you first of all, you check all of your equipment to make sure that the sound levels are exactly the same, so that the tone, is the same across days. Your cameras are set. You're putting the animals in the same boxes, running them about the same time every day so they're not too hungry or too full when you run them, and to just make sure all of that's consistent so that you get reliable data.
So this protocol, as you reported in this methodological paper, was previously used by your own lab in a previous research paper. What was the motivation to send the protocol to PLOS ONE to be peer-reviewed as a standalone methodological paper?
Yeah, so I really wanted to have some kind of formal protocol out there that outlined all the steps. I really wanted to make public to everyone that was interested the different kinds of materials and supplies that we used. Sometimes when I was setting up my own lab, I often found that I didn't remember, the kind of sucrose pellets that I used in my postdoc lab. Or, what is that little tiny thing that we had? I don't know where to buy it. I don't know, I don't even know where to look. So I thought that was really important to have all those things laid out in a single place so that other people can go and look and say, oh, I want to use this task for something. Let me see how they did it. And, they can get it up and running much more quickly instead of having to hunt around to find all these little obscure things that are important for the task.
And how did the feedback that you received from the peer reviewers shape the final version of the paper?
They were really important. There were a lot of details that I kind of didn't think about. So that was really helpful to get the feedback from the reviewers. There were things like thinking about if you're breeding your own animals, which I hadn't done before, are these partner animals from the same litter or are they from a different litter? And that's really important because if they are from the same litter, they might have some level of familiarity with each other. They might pick up on cues compared to animals that might be from different litters. Um, Also thinking about different details that were important for training in the different phases. So lever press training, are there different ways you can get the animal to the same endpoint of training before you start the conditioning procedures, and things like that. So it was really helpful to make sure I added in all those details that I normally don't think about, but other people might.
And my final question might be, a little bit speculative at this point because this is a relatively young paper that was published just a couple of months ago. But what do you think will be or has been the significance of getting a peer-reviewed protocol published for your career as an early career researcher?
It's been great. Just the process of writing out the protocol and getting this paper prepared and everything has let us think about this task in many different ways and consider, all the different manipulations we could do. What are some other ways we can change the task to look at, for example, if there are two partners of the opposite sex. So if we have a male rat with a female rat, what would happen? If we had animals from different ages, a young rat and an old rat, what could happen? If we have a rat partner that has a different experience from the other partner. So for example, if we have a fear conditioned rat with a naive rat doing this task, what might happen? So there's all these different questions that started coming up in how we can look at, different partners and how that affects their learning. Also different factors or measurements that we can look at. So for example, in this social setting, there might be some vocalizations that the animals are doing. So now we're starting to record the ultrasonic vocalizations during the task to see if that to see how those relate to the behaviors that they're doing during the task. And also incorporating new techniques. So not just, doing pharmacological inactivations. We're also looking at CFOS, immunohistochemistry, to see what areas across the brain might be activated in this task and how they compare to other forms of avoidance. We're also modifying the task in other ways. So now we're looking at observational learning of avoidance. So we can use the same setup, but have an observer rat watch a demonstrator rat do the task, and then we can test them later to see how observing the animal helps them learn avoidance. And then also trying to incorporate new techniques like electrophysiological recordings in the social setting, and things like chemogenetics, et cetera. So we're really excited to modify this even further.
Thank you so much, Maria. I don't have any other questions, but I don't know if you want to add anything that I haven't discussed so far.
I'm just really excited because it seems like there's a lot of new researchers using the PMA task to understand active avoidance and other behaviors in rodents. We're starting to see new papers come out using this task in mice, doing slightly different modifications with the reward and the motivation to get them to avoid. So I think this is a really exciting time to be looking at avoidance and also see where the field takes us in the future.
Thank you, Maria.
Mm-hmm.